Thank you. It's a real pleasure to be here. I wanted to thank Kevin for inviting me to speak here. I remember the very first Horizons conference I was at in 2007 in New York City, and I just could not believe that a conference on psychedelics was happening. So that was super exciting, and it's been great to see it evolve over time, and it's great that it's here in Portland for the first time. The historical research era with psychedelics was from the 50s to the 70s, and it was a big part of psychiatry, but something that I learned nothing about in my training. But during this period of time, there were over 40,000 research participants studied. The most studied drug was LSD, very little bit was psilocybin, and the most promising indication was the use of LSD-assisted psychotherapy to treat alcohol use disorder, followed by psychiatric and existential distress and advanced cancer. But the Controlled Substance Act hit in 1970, the war on drugs was declared, and psychedelics were buried from the history books. In the modern era, we've been studying psilocybin more than LSD, and today I'm going to talk about the three most advanced clinical programs with psilocybin, including the treatment of psychiatric and existential distress and advanced cancer, major depressive disorder, and alcohol use disorder. In terms of disclosures, I receive research funding from several sources over the last 36 months, including parts of NIH, such as NIDA. And I'm going to tell you about a really exciting grant I just received from the National Cancer Institute. But before NIH would fund psychedelic research, we've relied on nonprofit entities like the Hefter Research Institute, USONA, MAPS, and I also receive research funding from Reset Pharmaceuticals. I'm the director of the NYU Langone Center for Psychedelic Medicine, which is a training program which is funded by MindMed. And I'm listed as a co-inventor in two provisional patent applications for the use of psilocybin in advanced cancer to treat emotional distress. These were filed by my employer, NYU, licensed by Reset Pharmaceutical, and I've waived all rights and have no prospect of financial gain in relation to that. In terms of my background, I was born in Johannesburg, South Africa. I lived there for the first 6 years of my life. And at some point, things were getting difficult in the country, and my parents decided to leave. And there were three choices because my dad was in the diamond business. And the three choices were New York City, Houston, and Los Angeles. My parents made it to New York, and they absolutely hated it. The cabbie stole their luggage, and my mom said there's no way, which is too bad because New York City is my favorite city. They went to Houston. It was too humid. They arrived in Los Angeles, saw the palm trees, and my mom said, this is the one. So I grew up in Los Angeles, and it was an interesting time growing up in LA. And this was the very first time I learned something about psychedelics. Mostly my brother's friends and my brother were doing them a lot. And I was like, what is going on? Some strange activity there. But also, even though psychedelics in the 60s were centered in the Bay Area, there was a big amount of it in Los Angeles as well. And through the music and culture, I knew that something had happened, but couldn't quite locate it. Interestingly, I grew up in a town called Pacific Palisades, and I played a lot of sports, baseball and soccer. My favorite place to play sports was at the Aldous Huxley Recreational Center. At the time, I didn't know who Aldous Huxley was. Turns out, later found out Aldous Huxley spent his last years in Pacific Palisades. It's where he first did mescaline on the bluffs of the Palisades Hills. And he died there on the day that Kennedy was assassinated after his wife Laura had administered 100 mics of LSD. So there was this interesting history there. So I then went to college. I went to the University of Pennsylvania. And this was actually the first time I learned a little bit about psychedelics. In organic chemistry, we made these interesting molecules like LSD, at least sketched out how to make them. I majored in the biological basis of behavior, so in anthropology, I learned about the indigenous use of psychedelics. My favorite class was called Drugs, Brain, and Behavior, and they hinted about something about a guy named Timothy Leary and psychedelics. But that was sort of the end of it. I went on to medical school at UCLA, where I learned absolutely nothing about psychedelics, zero. I did my general residency training at Columbia, and a little bit here I'd heard psychedelics can make you psychotic, that they're psychotogenic agents. but nothing about their interesting history. Went on to do an addiction fellowship at NYU, and here learned about them, but again, learned that they were Schedule I drugs and basically bad. I've spent my professional career at NYU, all of it at Bellevue Hospital. I've been there 22 years. I came to NYU specifically to work at Bellevue because of its mission of helping the poor and underserved. And since I've been there, I ran a dual diagnosis inpatient unit for 5 years. This is where we treated mostly psychotic patients with substance use disorders and got to see people in really bad altered states and agitated sort of psychotic crises. I ran the Division of Substance Abuse for 12 years, a large program treating thousands of people with severe addiction and mental illness. And I ran our training program in addiction psychiatry. So I had a series of jobs, but interestingly, in 2006, had this amazing encounter with Jeff Gus. Jeff Gus was my supervisor at the time. One day he came into my office and said, what do you know about LSD and Albert Hofmann? And I said, I don't know what you're talking about. Jeff said that psychedelics had been part of psychiatry, and I started looking, and kind of hidden in plain sight was this just huge body of research. So I got very excited. Soon after that, Jeff, myself, and Tony Bosses formed the NYU. First, we were a reading group because we didn't think we could do research. But we formed a group, and we got mentoring from Charlie Grobe, and I've been conducting psychedelic research for the last 16 years, which has been a very interesting experience. The area where I think psychedelics can be the most helpful is in end-of-life distress. And psychiatric distress is very common in end of life. The treatments we have don't tend to work that well. And antidepressants, for instance, tend to do no better than placebo in people that have major depression in the context of cancer. Existential distress is particularly unique to end-of-life conditions and can be very common. Up to 50% of people have it. It includes this demoralization syndrome where you feel stuck, you feel you're never going to get beyond this thing, you sort of give up hope, have a hastened desire to die, and there's elevated rates of completed suicide in patients with advanced cancer that have this typology of distress. And when it comes to existential distress, we have particularly poor treatments. We have zero pharmacologic interventions. We have some existentially based psychotherapies, but they're underutilized and don't tend to work that well. So there's a huge unmet need to treat depression, anxiety, and existential distress in end-of-life conditions such as cancer, but it can be more broad than that. So this is sort of like a huge question: where do you want to die? And I think most people want to die at home with their loved ones, but unfortunately in American medicine, most people die in hospital settings. Our society has not done a good job of treating people at end of life. In my medical training, I was never taught anything about helping people at the end. It was you continue to treat them, never give up. And this, I remember doing the oncology wards and just seeing chemo given to the very end and thinking there's just something wrong here with this paradigm. So what does a good death look like? It includes reviewing your life in great detail, and we do this in the study, making sure that your pain is under control, and the importance of spirituality and meaning really comes out in end-of-life conditions. And it's important to have a sense of completion, to spend time with loved ones, make room for final dialogues, and to adequately prepare for death. And that's the thrust of the work that we did. So I wrote a review article a couple years on this topic, and the historical data was very interesting. It first started with a guy, Eric Cast. Cast was this amazing guy. He was both an internist and a psychiatrist at the University of Chicago. And he got this idea. He was a pain researcher. He got the idea. He'd heard about this drug, LSD, made by Sandoz. Back then, they were just sending it to researchers. And he thought, could this help my patient's pain? So he ordered a bunch of it up, and Cass started giving 100 mics of LSD to terminally ill cancer patients that had refractory pain syndromes. Just remarkable. No preparation, no nothing. I think, just dropping 100 mics in the morning and coming back later. It's just sort of the exact opposite of what you would think to do. But later, he, I think, had some people in the room. But astonishingly, he did this in 308 individuals. The group that came after that was the model we use today. And this was Stan Groff and the group at Spring Grove. This was sort of like the dream team. And what they did is they basically came up with this is medication or psychedelic-assisted psychotherapy, and that the psychotherapeutic container is key. that there are two therapists that deliver the intervention. There's extensive preparation before. There's extensive support that goes on during dosing sessions. And they were giving people LSD, so we're talking about a very long day. And then a lot of integration afterwards. A very skilled psychotherapy facilitated by these medications. And the model was totally different than what we're used to. Rather than one therapist, there were two. Rather than a 45-minute session, it was a 12-hour session. The couch was made into, the living room is made, the setting is made into a living room-like setting with a beautiful couch. And this is the model that we essentially have used today. And what Cast and Groff found, and this was pre-post because they did not have a control group, but that there were reductions in pain rapidly. He even did Cast a comparative efficacy study between LSD and opiates and found that both of them worked for 2 hours, but the LSD pain effects lasted for up to 12 hours in his first study. But in addition to that, Cast was the very first person to show that in addition to reduced pain, his patients were saying, I'm not scared to die anymore. I had these unusual philosophical insights. So that was the very first mention that psychedelics could help existential distress. And this research also suggested they have anxiolytic and antidepressant effects. But before they could go on to the control trials, everything escaped from the lab. Timothy Leary was thrown out of Harvard. The American public started using it. Psychedelics went from wonder drug in the 50s to demon drug in the 60s, and by 1970 they were banished, made Schedule I, and really sort of erased from the history books. A very dramatic thing that happened in drug history and drug policy. But in the last 20 years, the research has come back. And the work with advanced cancer, I think, is the sort of the most interesting. There was work that was done in Europe in LSD, and they've done work in advanced cancer and other advanced illnesses. And in the US, the focus has been on psilocybin, and there was a coordinated collaboration between UCLA with Charlie Grove, our group at NYU, and a group at Johns Hopkins led by Roland Griffith. The LSD work, I'll mention it a little bit. Peter Gasser did a pilot study published in 2014, 12 participants. Not all of them had advanced cancer, but they had other end-of-life conditions. And he was able, and these were 2 doses, 3 weeks apart. And he was able to demonstrate safety and reported on acute and sustained anti-anxiety effects up to 12 months. Matthias Lichty in Basel, teaming up with MindMed, just finished another study, bigger, N of 46, using 2 doses of LSD. And these are people that had both life-threatening anxiety and non-life-threatening. And this paper is under review, and they have some positive findings to report. So Jeff, Tony, and I, we never thought we'd get very far, but it turns out, like early on, I was able to get an IND from the FDA, got approval from them, and the DEA was signaling they would give us approval, but Bellevue Hospital said, there's no way we're going to let you give a psychedelic in this hospital. We're the hospital that's been, in one floor with the cuckoo's nest. Everyone thinks of Bellevue as the madhouse. Last thing we want to have them think is the place that we give psychedelics to poor people. So that really was upsetting to me, because this was like my home. And so I thought, well, that's it. I tried. Didn't think I'd get very far. But turns out the dental college at NYU reached out to us and said, you can do it here. So we conducted, I think, the only psychedelic study ever at a dental college. It was pretty amazing. That was the Bluestone Center for Clinical Research. They had a beautiful room, ensuite bathroom, and it was an amazing experience to do that there. Later, when we wanted to do the alcohol study that I'll mention, they said, well, that's too far. We can't have alcoholics walking around a dental center. You have to go back to Bellevue. And I said, well, we don't have the option to go back to Bellevue. But it turns out with some positive media and our publication, Bellevue took us back. So we are been back at Bellevue the last 7 years, and we are now centered at Bellevue, which is important to me because this is how I think we're going to get a more generalizable sample of patients that are not typically in research. So Charlie Grove published his paper in 2011 in 12 individuals with end-of-life cancer. And then early December 2016, the Hopkins group and the NYU group published 2 papers on the same day. Very similar design. And the design of the study here, let's see, does it have a pointer? Yeah, so, we took people that had life-threatening cancer that had anxiety and depression. There was preparatory psychotherapy over 6 hours before. We went into great detail the story of cancer and how it's negatively affected them. We embedded that within the story of their lives. We got them ready for the sessions, a lot around safety. And this trial was, it was 1 dose of psilocybin versus an active placebo, niacin. Then there was 7 weeks of integrative psychotherapy, and the primary outcome was here at 7 weeks. And then there was a crossover, meaning the niacin group got psilocybin group got niacin. Our primary outcome measures were anxiety and depression, but we also looked at existential distress, quality of life, and we measured the mystical experience. To give you a sense, this was not a nationally representative sample. It was more women than men. It was mostly Caucasian individuals. We were unable to recruit from Bellevue at that time. It was mostly women that had metastatic gynecologic cancers, and it was mostly people with advanced cancer. And mostly people that had adjustment disorder. They did not have a long history of mental illness. They just had this horrible reaction to cancer with a lot of existential distress. What we were able to find in terms of adverse events, this is very typical of psilocybin. It can increase pulse and heart rate, but it doesn't do it in a way that causes serious cardiovascular problems. There was some transient anxiety and psychotic-like episodes, so this can happen. People can have very anxious experiences, and how you manage it and help them get through it can be very therapeutic. But we had no serious adverse events, consistent with psilocybin being a very safe molecule. What we did find, so this is the psilocybin first group. You can see that when they get psilocybin, they have these rapid reductions in anxiety and depression. There's a big separation between the control group. And you can see that the effects here endure at 7 weeks post-dosing, and again, big separation between the groups, very statistically significant, and large magnitude of effect or effect size. So this was the strongest scientific part of the study. We were able to conclude that 1 dose of psilocybin was a rapidly acting antidepressant and anxiolytic that lasted 7 weeks. But at this point here, with the crossover, this group here got psilocybin. You can see their distress starts to come down and approach the other group. In total, we were able to, the findings suggest that a single dose works rapidly and lasts up to 6.5 months. And this was a really sort of interesting finding in the field, and something that we hope to replicate. In addition, we found that psilocybin reduced existential distress. We measured the demoralization syndrome and hopelessness in cancer. We found that it increased spiritual well-being and increased quality of life. We measured the mystical experience with the mystical experience questionnaire, and we found that psilocybin induced mystical states compared to placebo. We did what's called a mediation analysis to see if the intensity of the mystical experience here at 6 weeks post-dosing predicts reductions in anxiety and depression. And this pathway was positive, suggesting the mystical experience as a kind of candidate psychological mechanism of action. It's become a sort of big thing in the field. Does the subjective experience matter, and for which disorder might it matter? Gabby Egan-Liebes was in our group, and she designed a long-term follow-up study. And we collapsed all of the groups, so there's no between-group comparisons, and we just looked at people over time. And this was a 4.5-year follow-up. And what was interesting is that anxiety and depression continued to be in sustained remission all those years later. We cannot attribute that to the psilocybin, but it was interesting to consider. And we also had long-term reductions in existential distress like demoralization. The participants rated these among the most significant spiritual, meaningful effects of their lives. They said theirs up there with having kids. So highly memorable events, often having to do with confronting death and cancer. We did a secondary analysis looking at suicidality, and these between-group differences are not statistically significant, but we were probably underpowered to look at effects here. But you can see there's a trend that the psilocybin group tends to have more steep reductions in suicidality. And if you collapse the groups, you can see there is a reduction from baseline in all participants to 6.5 months in terms of reduced suicidality. And so it would be important to design a study specifically to look at that, because suicide is so common in cancer. So in sum, we were able to show that we could do this safely, that it was feasible, that psilocybin was both a rapidly acting anxiolytic and antidepressant, The clinical outcomes were very significant. There were sustained clinical benefits. Possibly there was anti-suicidal effects. We found reductions in existential distress, improved quality of life, and that these are among the most meaningful experiences of life. And the mystical experience emerged as a candidate mechanism of action. So this publication generated a lot of media attention and led us to want to do more in this area because it was so promising. And the questions to ask are, the data is early. You have to be very skeptical of early data. So what we really need to do is bigger studies to see if these are real effects. Does psilocybin and other psychedelics work for psychiatric and existential distress and advanced cancer? And if so, how? To also design studies to look at mechanisms of action, either neurobiologically or psychospiritually. But we don't think there's something unique about cancer end-of-life distress. We think that there are other medical illnesses where there could be that distress. So that body of research is moving forward. And another area is to look at cancer-related pain, and I'll very briefly mention something about that. So the resources needed to do this research, NIH did fund it, but they stopped doing it. It was impossible, so we had to use private philanthropy and nonprofits. But I think we're now at an inflection point where NIH money is now available, which is very important because it's the most unbiased form of funding. And at NYU, we are trying to conduct larger trials of psilocybin-assisted psychotherapy in advanced cancer, and wanted to tell you about some progress. So after 4 years of trying to get a grant from the National Cancer Institute, finally, about 2 months ago, I received a grant, an oral one from NCI, to study psilocybin-assisted psychotherapy in advanced cancer in 200 participants. It's going to be a multi-site study that happens between NYU and Denver. And I'm hoping that this can be a platform that can be used to consider how psilocybin may be available for this patient population ultimately. In addition to that, Charlie Grove is leading an effort, along with Tony Bossis at NYU and Brian Anderson at UC San Francisco, to sort of like go that extra step beyond cancer. And they are looking at the use of psilocybin-assisted psychotherapy to treat depression, anxiety, and existential distress in any end-of-life condition. I think that makes a lot of sense. Yvonne Boussant at Dana-Farber has started a pilot trial in this area. And so I think that's the sort of ultimate bigger goal, because the patient population is not just in cancer, it's in any end-of-life serious medical illness. Lastly, I'm just going to mention that we're going to be doing our first LSD study. I designed a trial of using LSD to treat metastatic advanced cancer pain. We're going to focus on bone pain. And I've been working with the National Institute of Neurologic Disease and Stroke on a grant that has made some progress. And I think this is an interesting area to go to. So I think ultimately if there's enough data for psychedelics and they get rescheduled, I think there has to be a robust regulatory framework. I think the FDA is going to have some oversight. It's really important that you have qualified therapists doing it. I think it's key to avoid bad things from happening and to optimize efficacy. And ultimately, if psychedelics are available, let's say psilocybin for end-of-life distress, you can imagine the settings of care that you would use them, such as hospices, palliative care centers, and cancer centers. So let's talk about major depression. Major depression is a huge public health crisis. This is one of the most disabling brain-based illnesses. Second most common cause of medical morbidity, associated significant rates of suicide. And our current treatments have limited efficacy and can have a significant side effect burden. And I'm going to review, how much time do I have left here? Just want to. Great. So this is an interesting area, and as I take you through it, I want you to consider that early pilot data can be sort of promising, exciting. The key is to do much bigger studies, because in the bigger studies is where the real truth emerges about whether there's a treatment effect. And I think major depression, psilocybin to treat major depression is a good case in point, because there's been more in this area than any of the areas that I'm going to mention. The first publication was done by Robin Carhart-Harris at Imperial. This was an open-label study of 2 doses of psilocybin in 12 individuals, administered 7 days apart. And Robin was able to show significant differences and improvements in depression from baseline to 1 week and 3 weeks. So this was a promising start. There was another study done at Johns Hopkins, led by Alan Davis, where 27 participants were given 2 high doses of psilocybin 2 weeks apart. And in terms of outcomes, this is 5 weeks corresponds to 1 week afterwards, 8 weeks corresponds to 5 weeks after. You can see there's a big separation between these 2 groups, and the effect sizes are quite large. So this made us think that this may be a very robust effect, maybe a similar on the order of what happens with cancer-related distress. They did a long-term follow-up here. This is collapsed across all groups, and you can see this is at 1 year. They were able to find sustained reductions in depression. So this was also promising as a finding. Robin Carhart-Harris published another study. This was a comparative efficacy study looking at 2 doses of psilocybin versus standard antidepressant escitalopram. It was in 59 individuals. This study was interesting in that this is the psilocybin group, this is the escitalopram group. These between-group differences were not significant. You see there's some, confidence intervals are overlapping. So, and the trial was also designed as a superiority trial rather than a non-inferiority. So they were unable to say that psilocybin was as good as the Lexapro. But it would be important ultimately to do that kind of study, because if 2 doses of a medication can achieve the same thing as a medication that you give every day for weeks to months, it has a significant side effect burden. Even if it works as well as typical antidepressants, it may have an added benefit of having this decreased side effect profile. Robin was able to find, it appeared that some of the secondary outcomes like anxiety and well-being that tended to favor the psilocybin group. But there was not correction for multiple comparisons, so these findings were considered exploratory. Compass now has done the biggest trial. They have given psilocybin to patients with treatment-resistant depression. They have breakthrough status for this. And I think they're getting close to publishing this data in 233 individuals. But in this study, it looks like the positive finding is here at 3 weeks, that after 3 weeks, the psilocybin 25 milligram group did better than the 1 milligram group. But it looks like these treatment effects disappeared here. So if psilocybin single dose works for 3 weeks in TRD, that is a benefit. Ketamine works about 1 to 2 weeks in this patient population. It's a very difficult to treat population. But so we're really going to have to see, as the data unfolds, where is the benefit and what does it really look like. So in terms of the future of this, an important next step is to see Compass's publication and to take a look at that. But the USONA Institute also has breakthrough status for the use of psilocybin in major depressive disorder, and they just completed a trial in 100 participants with major depression, single dose psilocybin versus placebo. We were part of that study at NYU, and USONA is sort of on pins and needles to find out what is the result. You do these studies after several years, and you have equipoise. You think your thing might work, but you're not sure. So the readout from this study will be really important. And if it is a positive study, USONA and Compass will be moving forward to phase 3 studies. So this may ultimately be the leading edge of psilocybin therapeutics for depression, but we'll have to see if the data works out or not. So the last thing I want to mention is the use of psilocybin-assisted psychotherapy to treat alcohol use disorder. Alcohol addiction is probably the worst of all of them. Alcohol is really the most toxic compound, even though it's legal. So it's interesting that alcohol, not a Schedule I drug, compared to psychedelics, that their risk profile is nowhere near alcohol. Is a big part of our culture, but it's a horrible compound. It causes all kinds of problems in the mothers and their children, It causes cancer, it can cause irreversible liver disease, it can cause irreversible neurologic illness, and under the influence of alcohol, people kill themselves, kill others. One of the biggest public health problems in this country, 100,000 people die every year from it. We have 3 FDA-approved medications. They work okay, but they tend to never be prescribed because people with addiction in this country are not viewed as sort of worthy of an integrated system of care. We do have some psychotherapies that work, but this is also a high unmet need in medicine and a place to develop novel therapeutics. So psychedelics are considered the most addictive drug, no medical utility, but it turns out they are not addictive in any way that we can measure them. They're not in animal models. You can't get animals to re-administer them reliably. They don't induce conditions place preference. The nucleus accumbens, the reward center, doesn't light up when you give them. And in humans, there's no such thing as LSD addiction where people use it every day and compulsively use it. It just doesn't occur. They are not addictive. In fact, they tend to have anti-addictive properties. And if you look at drugs, if you were like going to create a rational drug policy, you'd look at something like how lethal a medication is and how addictive it is. And you can see opiates, nicotine, alcohol, cocaine, these are the most damaging substances in society. The psychedelics are towards the least harm. So our policy is not a rational one. William James, interesting guy, wrote Varieties of Religious Experience, used nitrous oxide to sort of help get him in these states, which is an NMDA antagonist like ketamine. But he was the first to say people that find religion are cured of their alcohol problem. Bill Wilson, too, got sober in 1935 in Manhattan, not naturally, but he was under the influence of a psychedelic, a belladonna alkaloid. So he had the drug-induced mystical experience, the beginning of AA. And this is the part of AA you also never hear about. In the 50s, he undergoes 6 sessions of LSD treatment with Sidney Cohen at UCLA, finds them just like his spiritual conversion experiences, wants to introduce LSD into AA, and they said, no, Bill, we got to shut you down there. But it's interesting that 12-step, which is the biggest provider of treatment in the world, has these sort of mystical psychedelic roots. There was a meta-analysis in the 60s, and there were 5 randomized controlled trials, and there was a definite treatment effect of the LSD group versus placebo in terms of sustained abstinence. Michael Bogenschutz, who's the director of our Center for Psychedelic Medicine, started a pilot trial at New Mexico that included 10 individuals, gave 2 high-dose sessions of psilocybin in conjunction with motivational enhancement therapy and CBT. So very much the psychotherapeutic container we think is really, really key. And Michael and I thought that psilocybin would go very well with this type of intervention. In the pilot trial, there were rapid and steep reductions in drinking. And based on that pilot trial, Michael and I designed a much bigger trial that we just published in JAMA Psychiatry after 7 years, which was really exciting. We were able to randomize 93 individuals, 2 high-dose psilocybin sessions in conjunction with a 12-week psychotherapy platform. And you can see here, this is the design. So, there's 4 weeks of preparatory psychotherapy here. There's the psilocybin versus the control. There's more psychotherapy. There's another drug versus placebo, and more psychotherapy. So this is, a lot of psychotherapy here, and we think it's ethically important to provide that as a platform. This was also not a representative sample, more men than women, mostly white, Caucasian. So we want to change this in these bigger trials. We want to specifically include more individuals to make the results generalizable. In terms of what we found that both groups here are drinking about 50% heavy drinking days out of a period of time. And you can see here right before they get psilocybin, both groups have a rapid reduction in drinking. And part of this is you have to be willing to try to not drink before the session, and you're getting psychotherapy. And you can see here, though, the psilocybin group has this further reduction, that their drinking goes down, and the differences are significant. So the psychotherapy works for both groups, but the psilocybin worked even better. And it increased the drinking over the placebo group greater than 50%. The effect size was 0.52. We also found, this was dichotomous outcomes, that the psilocybin group had more abstinence, more non-heavy drinking days, and a greater reduction in risk level. So I'm going to end in 1 second here. The next stage here is that BMOR, which is the sponsor of this research, has just got an IND through the FDA for us to do a much bigger trial that will be led by NYU. 216 individuals with alcohol use disorder. We're hoping that this will be a pivotal trial that puts psilocybin on track to be made available for alcohol use disorder. BMOR is a nonprofit. The trial is going to cost $25 million. But this one's sort of lined up. And if it goes well, I think this could radically change addiction treatment. Could you imagine you go to a rehab because you have substance abuse, but at that rehab you're offered psilocybin as a way to help your addiction? Or even before that, MDMA. If you have PTSD as one of the core components of your addiction, MDMA is going to be available in rehab settings. So I think psychedelics have the ability to change the care delivery for all these huge problems in medicine in profound ways. So wanted to thank you very much for your time.
Thank you, Dr. Ross. It was wonderful to see the long trajectory of research that's been taking place in these arenas, so I look forward to hearing more about that in the Q&A. And our next speaker is Dr. ANDREA Gomez. Dr. Gomez is an assistant professor in the Department of Molecular and Cell Biology and the Helen Wills Neuroscience Institute at UC Berkeley. Her work aims to understand the instructive cues that sculpt patterns of brain activity. Her efforts led to the discovery of RNA-based programs critical for synaptic plasticity. The robust and widespread neuroplasticity induced by psychedelics motivates the Gomez Lab to decode the synaptic mechanisms that underlie cognitive flexibility. At the Berkeley Center for the Science of Psychedelics, Dr. Gomez is committed to advancing research, education, and training programs that properly acknowledge indigenous science from which present-day psychedelic practices stem. Dr. Gomez.
Guwatsi, Portland. I'm ANDREA Gomez, and as mentioned, I'm at the UC Berkeley Center for the Science of Psychedelics. I am a developmental geneticist and neurophysiologist, and I would say my scientific quest is to understand how we became so complex. What information in our genome, what information in our DNA encodes for how patterns in connections are organized, how patterns of activity, patterns of brain activity are organized, and how they propagate through neural networks. And importantly, what molecular coding principles balance order and variability in the brain, which allow for neuroplasticity and continued learning throughout life. And it is precisely this question about how neuroplasticity is organized that has led my research into the field of psychedelic biology. So I hope for today that I will provide a psychedelic biology 101 for everyone. In talking to Kevin Baltic in preparation, I wanted to kind of get an overview of what do we know about how these medicines work in molecular pathways, with how they work in cells and in neurons to potentially change activity in the brain.
Anything specific I want to talk about?
So as mentioned, I am affiliated with the UC Center for the Science of Psychedelics. It is an academic center founded in 2020, which is focused on psychedelic research, training, and public education. I won't go into detail too much about, but just to note that our work spans 5 main focus areas: public education and journalism, our research program, which includes human studies as well as studies from my end of molecular and cellular biology. We have a psychedelic guide training program, and I'm also happy to lead the diversity, equity, and inclusion efforts as well as reciprocity efforts that we have at the center. And I'd just like to make a note about some of these efforts, which I'm very proud of, is that indeed indigenous use of psychedelics goes back millennia, and the relationship of these compounds, of these medicines, and relationship to the growing interest from outside contexts has often led to cultural appropriation, cultural destruction, and environmental harm, most notably the mescaline psychoactive compound found in peyote. Another aspect is some of the relationships of how indigenous knowledge practices have been shared within Western science context. Most notably, Maria Sabina, the curandera of the Mazatec, who was able to share her ceremony with George Washington, who then went on to write an article that put essentially their local town on the map. Her local community was really discouraged by all these new psychedelic tourists, and she was essentially outcasted and died in poverty. Also, we note there's some notes from the Inquisition that described burning of the stake of herbalists from those who were using peyote, and they condemned anybody who would be using these compounds as heretics and to be punished as so. So in an effort to, I guess, develop some reciprocity for the ongoing research that continues, wouldn't necessarily be considered research in the Western perspective, but I would say that indigenous science research in the ceremonial form is ongoing. It's often referred to as ancient, but I would also say that it is contemporary as well. And so some of the efforts that we are doing at the center is to honor the indigenous principle of reciprocity. And this includes our first cohort of indigenous research student fellowships, which work to address issues of reciprocity and equity in psychedelic spaces, which includes the stewardship of plant medicines, public education, and journalism related to plant medicines and psychedelics. And this year was the first year of this fellowship, where we awarded 5 graduate students and 2 undergrads. And we have committed to 35 more research fellowships over the next 5 years. And one aspect that I am particularly tuned into, especially thinking about the Maria Sabina story, is how do we balance acknowledging and promoting indigenous contributions, at the same time protecting sacred knowledge. Indeed, our academic institutions are colonial institutions, and we have no intention of commodifying sacred knowledge, but we also want to develop a field that includes indigenous voices. And we think that developing is as a field in the form of our fellowship is a way to advance the field. And finally, another effort that I'm very proud of with the center is that we have decided to pledge 10% of new unrestricted funds to go towards DEI efforts. And I believe that this is a fantastic effort to start asking broader questions in the field, such as what are we trying to heal, not just in the scope of like fabulous clinical studies like Dr. Ross has discussed, but also kind of making relationships to our world and that we live in, the societies that we live in, and what are we trying to heal from. So I anticipate that many of you already seen this before. This was published a couple of weeks ago by the colleagues at Johns Hopkins, showing that in preparation for this psychedelic bubble that has been driven by this excitement by media and industry interests, is poised to burst, which they say that we are now at the peak of inflated expectations. And so what shall we expect, or what can we combat this bubble? How do we maintain the effort that so many people have done to try to get these medicines to people, and I guess allow for what strategies do we have? And they suggest that researchers, rigorous researchers, can provide some of the mechanistic substrate to kind of bring this back up. And part of this, the lack of the, I guess, mechanistic research just is exemplified by the fact that 1,000 clinical papers had already been published by 1961, but in a survey done last year showed that there have been 16 preclinical studies and 4 clinical studies to understand how psychedelics induce neuroplasticity. Now, some of the logic or evidence that there are long-term effects after a transient exposure to this medicine or a compound is that you see an immediate relief that endures for a long time. I'm not going to go too much into the clinical data sets, but this evidence from different depression clinical trials using ayahuasca, psilocybin, LSD, and shows that there are persistent, long-lasting effects in these patients. Yet, how does this happen mechanistically? So, some of the field's hypothesis for how this is happening is that our DNA, which, as I mentioned at the top, encodes for the instructions for plasticity, that in a normal situation, say a learning experience that induces the engagement of the DNA to produce new RNAs to then develop and integrate into neural circuits to change the function of those circuits. However, we hypothesize that psychedelics robustly engage the genome and induced a large-scale transcriptional response that can then go on produce the components that can change the function of neural circuits. And indeed, we know that and hypothesize that these psychedelic-induced plasticity RNAs are what's contributing to the elevated synaptic plasticity, the elevated cognitive flexibility, and some of the depression, anxiety, and addiction relapse reductions that we see from the clinical trials. So as I mentioned, I'm going to try to give you all a psychedelic biology 101. And I wanted to just kind of walk us through some of the core neurobiological features. And here I'm using the hippocampus, which is a structure located in the center of our brain that's critical for learning and memory. And when we think about network activity propagating through this circuit, and it does so in this general direction, we know that the activity is not continuous. In fact, it is interrupted as neurons pass the information from neuron to neuron throughout the circuit. Synaptic activity depolarizes the membrane, essentially elevates the membrane potential till it reaches a threshold. That neuron fires, elicits a synaptic current to the neighboring neuron, and so on and so forth. And this is why I'm so interested in synapses or the properties of synapses, because they have a profound ability to shape the way that information flow is engaged in an ongoing circuit. Now, I hail from a lineage of developmental geneticists that believe that the majority of our organization in the brain is predetermined. by select transcriptional programs engaged by unique cell types that create the components that will allow for unique propagations of network activity throughout the brain. And specifically, I'm interested in synaptic properties and in intrinsic properties, which I will explain in a moment. So the transfer of activity in neuron exists in two forms. And if there's a takeaway that I want you to learn today, this is really it, is that activity in the brain, or electrical activity, as we know, is occurring through not electrical activity from electrons, but ions. Sodium, chloride, calcium, these are charged ions that when they pass the membrane, they carry a current. And these currents have two forms. One is that's mediated by the synapses. When synaptic vesicle fuses with the membrane, they release neurotransmitters, and they can go on to bind to neurotransmitter receptors on a neighboring neuron, and then elicit current inside the neuron. And this is an example of a neuron that has been recorded, or a synaptic excitation event has been elicited. On the other hand, we have intrinsic activity, and this is the kind of the most famous type of activity of a neuron, which is the spike, or the action potential. And these action potentials are the form of rapid transmission across the neuron to the very end point of the axon, which then releases these synaptic vesicles. And then, as I mentioned before, this sequence of events repeats over and over throughout different networks in the brain. Now, there is a really important relationship between these two types of activity. And you can imagine that if you have too much synaptic activity or too little synaptic activity, then you don't get the proper propagation of these network flow throughout the circuit. Likewise, in intrinsic activity, if you have too less or too little, you fail to engage other networks of activity, and in a pathological state, if things are too excitable, you can result in epilepsy. Now, this back and forth relationship will balance themselves out. If we have overactivity in the intrinsic realm, we will see compensation by a reduction in synaptic activity. Likewise, if there is an elevation in synaptic activity, we will likewise see a reduction in intrinsic activity. And this balance is what's allowing our networks to maintain normal balance and for function. But interestingly, these balances do get shifted, and they get shifted particularly during learning and memory. And so how is this balance, and more importantly, how do psychedelics influence the input-output functions of neurons? This is a core focus of the questions that we ask in my lab, and to kind of highlight the reasons why is that we know that in, say, a mind-altering state, that indeed neurons are elevated in synaptic excitation. But how is it different from normal activity? How is it that the psychedelic mind-elevated experience doesn't completely disrupt this network activity? So I'm going to go into a little bit of what we know. So here is a list that many of you may have seen of some psychedelic compounds. Here I put a molecule of serotonin, and you can see that the tryptamines, which most resemble serotonin with this benzene and the pyrrole. Here you can see here psilocybin has a very similar structure to serotonin, likewise DMT. The other classes, the ergolines, which is the LSD, and the phenylethylamines, which mescaline, and also another important drug called DOI, which is actually the most well-studied psychedelic in neurobiology. Now, we know that these different medicines have different affinities to different receptors in the brain. You may have heard that psychedelics are binding to the serotonergic family. Indeed, this is true. But you can see that DOI, LSD, DMT, psilocybin, they have kind of salt and pepper binding affinities to receptors throughout the brain. And just to kind of show the specificity, here we have THC, and indeed it is very selective to cannabinoid receptor. Mescaline has a very more tighter binding affinity profile in the serotonergic family, but just know that these compounds do bind to serotonin receptors in addition to dopamine receptors and adrenergic receptors, amongst other type of molecules in the brain. And it is believed that the kind of this combinatorial activity is what's contributing to the subjective or the differences in subjective experiences amongst these different compounds. So some of the known structural changes that can happen after exposure to psychedelics. This is some studies from David Olson's lab, which look at the treatment of various psychedelics in cultured neurons. And you can see that after 72 hours in a dish, mind you, that there is this elaboration of these structures, these dendrites on the neuron. Likewise, you see these little protrusions, which are called spines. These are the sites of synapses. We can see that there is an elevation in the number of these spines or these synapses after treatment with various psychedelics. So this is a pretty substantial evidence that they are inducing structural changes in neurons. The last structural change that I will discuss is a study done in Alex Kwan's lab, which shows that after a single high dose of psilocybin, and you look at these spines, and as I just told you, that spines are the sites of excitatory synapses, that you can see that there is an elevation in fold change 30 days out. This means that after 30 days, there was an elevation of the number of synapses long-term in the prefrontal cortex. And here, this is now note this is in mice, but what you can appreciate that if you split the mice into female and male groups, that the large driving force of this result is given actually predominantly by the female mice cohorts. And another consideration is that these are neurons from layer 5 in the prefrontal cortex. So these are animal studies, and it's hotly debated whether or not animals are having hallucinations. We know that our brain and mouse brain are significantly different in their cognitive capabilities. But the one feature in mice that is reproducible for an induction or a specific psychedelic effect is this head twitch phenotype, which can be kind of likened to like a dog shaking water off. And it literally is called events per minute, like head twitches per minute. And you can see the different types of psychedelics, DOI, LSD, psilocybin, elicit these head twitches compared to vehicle. Now, what you'll see here is this plus plus and these minus minus sections. And you can see in this minus minus section, it actually refers to mice that had their serotonin receptor deleted. These are 5-HT2A receptors, and you can see that without these specific type of serotonergic receptors, that you lose this head twitch activity. Likewise, in humans, if from Katrin Preller's study, looking at giving humans ketoseronin, which is an antagonist of 5-HT2A receptors, that you completely lose the subjective psychedelic experience. Some more evidence that the serotonin receptors, specifically 5-HT2A receptors, is playing a significant role. We can see that the 5-HT2A receptor is highly enriched in the prefrontal cortex. And whenever you attach a psychedelic and a heavy labeled radioisotope and put it on a mouse brain, you can see that the psychedelic is localizing to the prefrontal cortex here in the mouse brain, but not in a mouse brain that has a knockout or has that 5-HT2A receptor deleted. So just to kind of go back to this idea that synaptic and intrinsic activity are playing this role, what does the physiological changes look like? Now I've just kind of discussed what some of the structural changes are looking like. Again, how does this influence the transcriptional responses or the response from the DNA? And some of the evidence showing here that, pardon me, from some original study from Javier González Maeso, looking at a microarray data. The best way to look at these are individual genes that were turned on in animals that were treated with DOI or LSD, and you can see that you get this large upregulation of genes that are completely absent whenever you have a 5-HT2A receptor knockout. And some later studies looking at how these genome-wide affect things, Javier González Maeso showed that indeed there are, the best way to read this data set is that a single dose of psilocybin of DOI was given at day 0, and then the genes were looked at 24 hours, 48 hours, and 7 days. And so what this is saying that in these different classes these are enhancers actually, that you can see that some enhancers get reduced over time, some turn on and then kind of go reduced. So you're getting these different reactions or different responses that are not just activation or not just downregulation, and furthermore they have different temporal profiles. Now, what is going on after 5-HT2A signaling? If we kind of take a look at where these are transmembrane receptors, they sit at the membrane and they bind to serotonin or a psychedelic, and they can elicit different intracellular responses. Notably, it can increase sodium channel transduction, calcium channel. You can release calcium from endoplasmic reticulum. You can change translation. You can change some precursors to endocannabinoid signaling. And the main point that I want you to take away from here is that it's extremely messy. There are a lot of things going on, and we know that neurons, different types of neurons in the brain, have different components. So it's likely that different neurons that have 5-HT2A receptors are engaging in parallel cascade signaling, molecular signaling events that then go on to engage their genome. So the next, I want to talk a bit about the physiology. Now, what does normal serotonin do in the brain? And here we have a recording from a cortical neuron in the prefrontal cortex. And you can see that if you apply regular old serotonin, you see it greatly increases synaptic excitation. However, oddly enough, whenever you, and this is just spontaneous, you're just recording, just listening to these events fluttering in the electrode. And physiologically, what's happening is the synaptic vesicles are randomly fusing with the membrane, releasing the neurotransmitter, binding to the neurotransmitter receptor, and then causing these currents on the postsynaptic cell. However, if you physically stimulate the synapse, compared to baseline, serotonin actually reduces the amount of evoked stimulation, which is a bit of a paradox. How is it that you have all this kind of random elevated synaptic vesicle fusion with a reduction in synaptic evoked excitation? And these researchers went on to take a look at what potentially could be happening. And what is essentially happening is that serotonin is increasing asynchronous release. Now let me walk you through what that means. So in a basal state, you can see this here. You see this little spike here. That's whenever you have a stimulation, and then you get a recording of synaptic currents. After the application of serotonin, 5-HT, you see all these little blips that are happening. And this means is that serotonin is essentially decoupling the synaptic vesicles from fusing during the time when it receives the information. So you're essentially creating a longer time that synaptic or other inputs can have a meaningful amount in relationship to this original stimulus. And whenever the treatment with DOI, which, as I mentioned before, is related to mescaline, we can see that not only does this evoked excitation get elevated, but you also have all of this extra fluttering of asynchronous release. So the best way that I think about it is that in a normal synaptic transmission event, that you have an event it goes through, but during a psychedelic event is that you are in a neuron that has been treated with psychedelic, is that you have the original event, and then you have all these excitatory echoes that are happening, which could contribute to altered function of neural circuits. I'm just going to kind of go through and just show that there are also other evidence to show that there's elevated synaptic excitation that occurs when treating with DOI, DMT, and that just the main takeaway here is that psychedelics are acting on these evolutionary conserved GPR systems. These are the serotonin receptors, that a single high dose can induce neuroplasticity as measured by changes in structural features of neurons, and that psychedelics induce distinct transcriptional events in a 5-HT2A receptor dependent fashion. And then finally, that psychedelics can increase synaptic excitation and transmission. So with the last remaining couple minutes, I will discuss some of the work that we are doing in my lab, and which is to kind of follow up on this idea that there is a large-scale plasticity event that's being responded to by the genome. And we actually don't know which of these responses is contributing to the long-term events. For an example, some of the genes that we work with, the synaptic genes, they are 1.3 megabases in length, which means that it takes 12 hours just to transcribe, just to make. And this would fall way long after the end of a psychedelic experience in psilocybin, for an example. So what kind of responses get turned on during the trip, then gets consolidated to solidify whatever type of motifs or ensembles of neurons that were activated. And the way that we are attempting to approach this question is, one, we ask the question is, what can psychedelics teach us about neuroplasticity? And then two, can we mimic psychedelic-induced plasticity? And indeed, I'm at UC Berkeley, which is the birthplace of CRISPR technology, the gene editing technology. So we are adapting CRISPR technology to validate the genes that get turned on and prove that they are indeed playing a role in plasticity. And just to briefly show you what the experiment looks like, we have a large-scale sequencing experiment ongoing. We're actually this week submitting samples, which is to look at 3 different sequencing technologies. The first one is called ATAC-seq. So if many of you may recall that our DNA is wrapped around these proteins called histones, and there can be little chemical modifications on that histones that relax the DNA or wind it up. And if a DNA is relaxed or whether or not it's wound up will determine whether or not it has a likelihood to be made. Next is called SLAMIT-seq, which is a technology that we can use to label newly synthesized RNAs and then assess what genes and what gene families get turned on immediately. And then finally, we can look at RNAs that are engaged on the ribosome. The ribosome, as you recall, is the molecular machinery that turns the RNA into proteins. And from this, at 4 different time points, we can get a really high resolution vision of what's happening, not just at the genetic level, at the newly synthesized RNA level, but also, what is being made into protein. We then pair that to our electrophysiological recordings in the prefrontal cortex and quantify different parameters that are maybe contributing to these long-term plasticities that we can observe in cognitive behaviors. And the way that we do that is we use a cell type specific strategy. We focus on the prefrontal cortex of mice. Indeed, mice and humans have vastly different brains, but the prefrontal cortex in mice are evolutionarily conserved for playing a role in cognitive flexibility, social cognition, attention, visceral pain, social rejection, and in humans, we know that there is overactivity in patients with depressions, feeling of connectedness, and also in both humans and in mice, it is enriched in serotonergic axons and serotonergic receptors. Here's a viewpoint of what the mouse prefrontal cortex is, and here we take this little chunk of the prefrontal cortex, and here is our cell type specific strategy. We can label different types of neurons in the brain using molecular genetics. And you can see that in the layer 2-3, layer 5, and PV interneurons, we can reliably label with our genetic strategies. And the last bit of data I will describe to you is the majority the majority of RNA sequencing data sets will just take a chunk of this tissue and then sequence it and see what happened. But as you can appreciate, the brain is made up of many different cell types that have very distinct functions, which are engaging in not just plasticity, but normal functioning of the brain. These green cells here are these excitatory neurons. These purple ones are inhibitory neurons. And this balance of excitation and inhibition is critical for how activity is propagated and regulated in the network. And the last thing I will show you is that we use a technique where we can specifically label excitatory neurons or inhibitory neurons. This is a demonstration of that, where we can pull down ribosomes that have RNAs attached only from inhibitory neurons. And this is a demonstration that we can enrich in an inhibitory marker and de-enrich in an excitatory marker. And when we look at brains that have been treated by a DOI after 48 hours, we can see a lot of genes known to go up after psychedelics, and this is in the entire brain whole cell lysate, whereas specifically in these inhibitory neurons, you can see these genes going down. And these genes are BDNF, which are growth factors. We have some synaptic genes, which is giving us an indication that plasticity is engaging on different temporal scales in different types of neurons, and likely playing a role in consolidating and engaging in different patterns following a high dose of psychedelics. So with that, I want to thank you for your time. I just want to also mention some of the other questions that we ask in my lab. We are also interested in the plasticity of the enteric nervous system. Indeed, where 500 million neurons line our entire digestive tract and is also the source of 95% of the serotonin in the body. We're interested in this relationship between the mind and the body and how plasticity and the molecular programs that are engaged. And we're also interested in developing and mimicking some of the psychedelic-induced plasticity to potentially identify therapeutics in conditions where psychedelics use is not appropriate, such as individuals with on the schizoaffective spectrum, or in situations like neurodegeneration, where a chronic use of psychedelic wouldn't be ideal. And with that, I would just like to thank my lab group, my young group of future psychedelic researchers. I'm very grateful to them and their hard work, and again to the organizers for inviting me to give you a talk.
Thank you so much, Dr. Gomez. It's refreshing to see methodologies that can look at the impact of psychedelics on the brain above and beyond just subjective questionnaires of mood and other psychiatric symptoms. So I appreciate all of that. Not that there's anything wrong with that, but it's nice to see all of the variety that you've been working so hard to put together. So now we're going to enter into a question and answer period. So for those of you who don't know how this works, there is an app that you can scan on your badge that will take you to a place where you can enter questions. So ideally, you're doing that throughout the talks so that I can get up here and look at my phone and choose some of the top selected questions. But before I do that, I'm going to ask a couple questions of my own. First, Dr. Ross, I'm curious. So we've talked a lot about the challenge of generalizing the results of clinical trials to real-world settings. And as you probably know, kind of what we're doing in Oregon is a bit different than what is done in clinical trials. So I am curious to hear your thoughts as to whether or not there's anything we can take away from the findings of the various studies of psilocybin therapy that you've been engaged in that would generalize to the setting and benefits that clients could receive in Oregon.
Yeah, thank you for that. Well, I think Oregon's about to embark on a very interesting experiment. I mean, it's sort of a groundbreaking drug policy in a way, and thing that's happening. I think the most important thing is to make sure that it's done with safety, and if we could impart anything from the way we do it, it's very much optimized for safety, that you have skilled care providers who are doing it. There's a lot of screening that goes in. Psychedelics are definitely not for everyone. Dr. Gomez mentioned psychotic disorders, for instance, or people that are at risk for that. Really, these drugs can induce psychotic states. And there, can be medication interactions that can be problematic. So I think safety is the most important thing. Screening people very robustly is very important. Making sure you prepare them for sessions in a very sort of intensive way, spending many hours getting to know whatever their presenting problem is, and then taking a life history. And then adequate support during sessions, and this needs to be done in sort of like the most ethical way. These are people that are highly, highly vulnerable states. They have, difficult conditions, and they're really made vulnerable by these drugs. So boundaries are going to be really important, high ethical standards. And then what happens afterwards in the integration, I think, is critically important. And again, having people that are skilled in knowing what to do with difficult material and people that have difficult problems. But the real world part of this is really going to be interesting to track, and we're going to learn a lot from this model, and the way it's designed is very thoughtful. So I'm looking forward to seeing how it evolves and what we in the science community can learn from this.
Thank you for those thoughts. I know we've spent many a long hour here in Oregon discussing these very things, so I'm excited to see how it will all turn out. Dr. Gomez, early on in your presentation, you asked a question that really stuck out to me and felt really poignant. And that was, what are we trying to heal from above and beyond, indications, mental health indications? What are we trying to heal from? I'm wondering if you could say more about that, and also how we might reliably measure whether this healing has taken place.
I think the metrics for whether or not we feel healed or not is still out for debate, but I view those types of questions as a scientist in the way that it's actually, I don't know the answer to that. But being at a public academic institution like UC Berkeley, I hope to recruit, smart young individuals who are also interested in kind of evaluating the relationship of these medicines to not just access our equitable access, but what does it have for, are the problems that are causing issues going to be persisting even if we have this? So are we really going to be healing or temporarily having a respite? And being able to evaluate the social and political contexts around, mental health care, I think is a very valuable venture to be thinking and having our young, bright minds being engaged in those conversations. So I guess maybe the short answer is I hope that becomes a field that we can develop at the center.
Thank you. Okay, I'll start reading some of the top-rated questions here. Dr. Ross, I believe this one is directed at you. Can psychedelics treat opioid disorder? And what are the contraindications for people using methadone or buprenorphine?
Psychedelics, there are several groups looking to use psychedelics to treat opiate use disorder. There's a group at University of Madison, Wisconsin, led by Randy Brown, that's testing psilocybin as a treatment in people that have opiate use disorder who are on buprenorphine. There's maintenance medication for people with opiate use disorder. There's opiate maintenance treatments like methadone, buprenorphine. There's opiate antagonist treatment with naltrexone or Vivitrol. And they tend to work well if they're used, but there's definitely room to develop other treatments for opiate use disorder. There was, LSD was mostly studied for alcoholism in the 60s, but there was one study looking at heroin addiction, and it was unclear what the results were. Our group at NYU is interested in this. Michael Bogensuch has designed a pilot study of psilocybin to treat opiate use disorder, and he's thinking of doing it in patients on Vivitrol, extended release naltrexone. We don't know what the drug-drug interactions are, so I think the first thing is it's safe to do in this population. It's unethical to do a trial of psilocybin versus placebo in people with opiate use disorder because over 100,000 people died last year from an opiate overdose, and we have very good treatments that work that we are not utilizing. So I think this is a really promising area, and there are other groups that have developed protocols to go after it. I mean, the opiate epidemic is horrible, continues to get worse, and finding new ways to treat it is going to be key, and I think psychedelics could be a part of that.
Thank you. Dr. Gomez, you mentioned that DOI is the most researched psychedelic. Folks are wondering, what is that, where is it found, and why aren't we hearing more about it?
So DOI has an interesting story, and the reason why it has been most widely researched drug is because it is a psychedelic and it is non-scheduled. And this has allowed researchers to use this drug without acquiring a lengthy DEA license for Schedule I use. In fact, that's part of the reason why I'm very interested in generating high-resolution data sets comparing DOI and psilocybin, because the majority of researchers and the majority of research institutions, I'm very fortunate to be at an institution that can accommodate a DA1 license. But I would say they're expensive, the drugs are expensive, the process of applying for is lengthy, and so already in just getting access to Schedule I drugs to do biological neuroscience research, there's already equity issues. So I think that the DOI studies will be helpful because they do operate on different pathways. I do know, not by experience, but that DOI has a kind of a different type of trip. I think that lasts 12 hours, and it was synthesized, I think, in the early part of the century. But yeah, and recently the DEA tried to get it scheduled this summer, but thanks to the community who wrote and said that, no, this is a very important research compound, the DEA decided not to make it a Schedule I compound, so that was a great win for the psychedelic research community.
Thank you. This next question, I think perhaps you both could answer. Curious your thoughts on whether or not a mystical experience is necessary for a client to experience healing benefits.
Did you want to start?
Yeah, I think there's not evidence out there yet that suggests that you can go without it to have the beneficial. I know that there are active developments to try to make non-psychedelic psychedelics, which I think would be the experiment to test that hypothesis. I would like to see the data. I would say that as I alluded to in some of the talk, that the type of activity, the synaptic activity that's engaged during a psychedelic exposure acts very differently than normal serotonin, for an example. And this kind of overactivity state, I would say, which is reflected in, I guess, the mind-altered experience, is probably critical for triggering unique transcriptional programs that could be then consolidated, or rather, the way that I envision it is that we have our normal brain ensemble of neurons, and then our psychedelic brain, and a unique other ensemble of neurons gets activated, and those induce transcriptional molecular programs that then consolidate that altered state. So I think it's a hypothesis that would be worth testing.
Thank you. Dr. Ross, do you have anything to add to that?
Just to add briefly, there's thinking going back to the 60s that this mystical experience was, a lot of people thought it was key to therapeutic benefit. And there have been trials that have definitely found associations between intensity of that experience and some clinical outcome. And our cancer study with psilocybin and the Hopkins one did do a mediation analysis to look at mystical experience and outcomes and found an effect. But that certainly doesn't prove causality. It's preliminary, and I would agree with Dr. Gomez. We need to test it, and we need bigger studies to take a look at it. And we need to be open that benefit may have nothing to do with that experience. But it's created this kind of like weird schism in the field. Some people are totally behind this is what's doing it, and other people, perhaps biological reductionists, think that it has nothing to do with that it's, a completely neurobiologic thing like neuroplasticity. And I think for sure with like end-of-life distress, the quality of these experiences I think is important. Our participants had experiences that had to do with confronting their death or confronting cancer, really important salient things that helped them, and they were aspects of a mystical experience that was associated with that. So I think more research, and the jury's still out, and we'll have to see.
Thank you. Next question, can you discuss the neuroplasticity potential of psilocybin to benefit people with Alzheimer's?
So Alzheimer's is a neurodegenerative disorder. The neurons are dying. And part of what's happening during that death process is that the circuits, or the neurons that remain, have to attempt to compensate for the lost parts of the circuit. And one appealing hypothesis is that if you could engage the remaining neurons that are present, and enhance their neuroplasticity, that maybe they could compensate a little bit more. Now, the dying part of neurodegeneration, I think it's a little bit unclear about what mechanistically neuroplasticity could play a role. I mean, if the neuron is sick, what would increasing synaptic strength do, or what would increasing the number of times that it can fire an action potential? I'm not sure. But I do think in terms of kind of a therapeutic way to kind of compensate or boost plasticity in the remaining neurons, I think is an appealing hypothesis.
Thank you. Dr. Ross, assuming that the upcoming NYU alcohol trial goes well, how long would it be until the FDA could make psilocybin available for treatment?
Well, I think, yeah,
Crystal ball.
That's an interesting question. I think between that trial, the work with psilocybin and depression, and the work in advanced cancer, I think one or all three of those eventually could lead to psilocybin being made available. Regarding the alcohol trial, I mean, we're going to start that trial early next year. It's going to be, dozens of sites, 216 people. If we complete that in 2 to 3 years, and if the division at FDA that deals with addiction products likes that data and considers it a pivotal trial, psilocybin could be rescheduled maybe 3-5 years from now, I would predict. But the trial may not work, and so I think we just have to see what happens next. But if it does work, then the implementation part of it is going to just be very interesting to make sure it equitably reaches all patients that could benefit.
Seems like a reasonable estimate, and I agree. It'd be lovely if we could just figure out what those timelines are going to be, right? But that was a reasonable guess. I think we have time for a couple more questions. How are we doing on time? Yeah? I'm short. I can't see the time clock without stepping on my tiptoes. Okay, so let's see. Dr. Gomez, are there other known drugs that provide neuroplasticity or that promote neuroplasticity other than psychedelics?
SSRIs. Trauma induces plasticity. Plasticity is a built-in function of neurons. Plasticity is often viewed as has a positive affectation, but decreasing synaptic strength or decreasing the firing of neurons is also plasticity. So I'm agnostic on the term plasticity. I think of more as a negative or positive magnitude value. But the most critical, I guess, perspective on it is how is it changing the function of circuits for particular outputs or particular behavioral changes.
Great, thank you. And also, is there any research on the effect of psychedelics on the gut-brain axis?
That's what we hope to find out.
I look forward to those results. Dr. Ross, I believe you remarked that you hope the future has a robust regulatory framework. What does that framework look like, in your opinion, for psychedelic therapy?
I think there has to be some structure that oversees it to make sure that there's quality control and that it's done in a kind of safe, ethical way. One way that the FDA may do that, the FDA does not govern the practice of medicine, but they, through their risk evaluation mitigation service, a REMS program, could stipulate, for instance, let's say MDMA, psilocybin can be given, but they have to be given with two therapists that spend X amount of time before, during, after. So that may be one way to do it. The Joint Commission is a regulatory body that audits hospitals to make sure that standard of care is being delivered. They could be another regulatory entity that makes sure clinically that there's correct oversight. And there are probably other ways to do it, but I do think it's important that we develop best practices and we make sure people are doing them and put it in line with other treatments that are delivered in the mental health field.
Great. Okay, one more final question. Dr. Gomez, the dynamic timing of induced plasticity seems to underline the importance of integration. Has anyone made this connection, or is this something your lab is looking at?
Yeah, I think that's insightful. That's the motivation for doing the specific experiments that we're doing, specifically the looking at how different cell types are engaging. I'm reticent about integration in like kind of the human perspective, and we are working with mice, but we are actively working to identify mouse behaviors, specifically cognitive flexibility behaviors, that could potentially be reflective of that maybe integrative-like feature. But I think the temporal aspect is critical. For an example, excitatory neurons, which spike at 8 to 20 hertz or 8 to 20 times a second. PV neurons, inhibitory neurons, spike about 100 times a second. And these engagements of these patterns, if they're shifted slightly, you can imagine could generate different types of temporal patterning. And by just marginally strengthening synapses on these connections, or marginally changing the magnitude of spikes that you can have in a neuron, could really robustly change how network function can occur. And indeed, it's considered some of these, many of you have heard of critical windows, these periods in development that have a very tight time window when they're closing. This is an example in social behavior. This is an example in vision or in speech, that if you don't get at a particular time point, then you lose that window. And it's known that this window is actually controlled by these fast spiking inhibitory neurons, their maturation. And so one maybe pet hypothesis would be that these kind of restrictive aspects that these highly fast spiking neurons have in controlling the network, putting the brakes on the network, maybe they're released temporarily. And wondering, how long is that temporal window? We are really excited to investigate as potential plasticity windows.
Great, thank you. And thank you both so much for sharing all of your insight and wisdom and experience with us, for your really detailed talks. I learned a lot. Appreciate you both coming out here to speak to us.